All right, I just wanted to give you guys a quick week 2 lab quiz to review. So we went over like exercise 5 in the lab manual, but this quiz will also talk about like other processes such as transport and diffusion, epithelial transport, protein interactions, tonicity, um, and osmolarity. Right, so I just wanted to hit on those topics with you just briefly. So remember the difference between passive and active transport processes. Passive processes do not require ATP or energy, right? Whereas active do require ATP, right? So let's go to passive processes. So passive processes, they don't require energy, right? And so these would be examples of diffusion. So simple diffusion or even facilitated diffusion, right? Simple diffusion is where a molecule is just simply going down its concentration gradient, right? We're going from an area of high concentration of solutes to an area of low solute concentration, right? And remember, diffusion is talking about the solute, right? And so it can either simply diffuse by itself or it can be facilitated. So facilitated diffusion is right where we're using like a transmembrane or a membrane protein that's helping this substance move down its concentration gradient. Like, so we mentioned like a GLUT transporter, right? That simply transports glucose. Again, it can transport glucose down its concentration gradient from an area of high glucose concentration to an area of low glucose concentration, right? So again, these would be examples of diffusion. Now, there are some things that we talked about before where, you can, modulate diffusion, right? So you can speed up diffusion. So diffusion will increase, if we have like high temperatures, for example. So remember that, okay? Increased temperature will have increased diffusion, right? Diffusion is quicker with smaller molecules rather than bigger molecules. And diffusion is much quicker, when you're closer to that molecule, and it's slower, with distance, right? Diffusion is powered by the stored energy, right? And so that stored energy is basically that potential energy. So remember, stored energy is that potential energy. Right? So for example, we use the stored energy or the potential energy of sodium to pump glucose against its concentration gradient, for an example of those SGLT proteins, right? Or even remember when we talked about oxidative phosphorylation or the cell respiration, right? When we looked at the electron transport chain, right? We were pumping hydrogen up to one side of the membrane of that mitochondria. And then it wanted to go down its concentration gradient. And we used that potential energy of that hydrogen ion to shrink that ATP synthase molecule to make ATP, right? So remember, stored energy is like the potential energy. And of course, the opposite, remember we talked about kinetic energy. And so kinetic energy is like the energy of motion, right? Things like that. Now, when we're talking about transport processes, right, we're transporting it across our membrane. Now, there are characteristics that, you have to remember and think about when we're talking about moving across the cell membrane. Now, larger molecules, right, they're not going to want to go through your cell membrane, right? Charged molecules, they're not going to want to go through your cell membrane. Okay, so remember those. Okay. When we're looking at active transport, so now active transport requires ATP, and so there's some examples of active transport. We have primary active transport, so like that sodium potassium pump. That's an example of a primary active transport where we're using energy directly. So we're using ATP directly. Or it could be indirect or secondary active transport. And again, this is where we're using a concentration gradient, okay? Now, vesicular transport, right? This is where you're transporting something in a vesicle, right? And so exocytosis is spitting out of the cell. Endocytosis is bringing something in the cell, right? And phagocytosis is basically cell eating. Again, we're bringing something in the cell, Okay, so remember these different types. Now, when we're looking at a membrane, right, we have two parts of an epithelial cell. I should say when we're looking at the cell, I should say an epithelial cell, right? So when we're looking at this epithelial cell, right, and we're looking at epithelial transport, so certain molecules, right, they can be transported paracellularly, so through or between, right, the cells. This is between the cells, or it can go transcellular, and that's through the cell. And so it has to cross over the apical and the basolateral membrane. So remember, apical membrane is also a mucosal membrane. It faces the lumen of that organ, whereas the basolateral membrane is the serosal layer. And so that faces the extracellular fluid. Okay. Remember these different type of protein interactions. So enzymes are types of proteins. These are biological catalysts. They speed up chemical reactions by simply lowering the activation energy required to start the reaction. Membrane transporters. So proteins can act as membrane transporters. These are proteins in the cell membranes that help to move substances back and forth between the intracellular and the extracellular spaces. We have proteins that can act as signal molecules. They can act as hormones, right? Or other signaling molecules that can, similar to like regulatory proteins that can turn the cell processes on or off or up or down. Proteins can act as receptors. They can bind signaling molecules and initiate a cellular response. And we also have binding proteins, right? And so these are going to be found mostly in the extracellular fluid. They're going to bind something. They're going to bind something and they're going to transport it, right? So albumin, for example, it binds to those steroid hormones and they transport them throughout the body. Or we have hemoglobin. Hemoglobin, right, binds oxygen and carbon dioxide and transport it throughout the body. We also have immunoglobulins, or these gamma globulins, or these antibodies, right? These are extracellular immune proteins that help to protect the body against foreign invaders. And we also have structural proteins that help to form cell junctions and form the integrity of that cell membrane. Remember tonicity, right? So we have isotonic, hypertonic, and hypotonic solutions. So it describes when the cell is placed in a certain solution, what will the response be? So if a cell, red blood cell, right, is placed in a isotonic solution, it's going to have the same amount of stuff inside the cell and outside the cell. So water will move in and water will move out equally. So in an isotonic solution, there will be no cell shape change, right? So the shape of the cell will remain the same. If you place that cell in a hypertonic solution, right, and that's where the solution has more than the stuff than the cell does, water wants to move where there's stuff at, and if there's more stuff outside the cell, water's going to move out of the cell. And that cell will basically shrink up and crenate, right? And so in a hypertonic solution, that cell will crenate, shrivel up and die. In a hypotonic solution, there's less stuff in the solution than the cell. So if there's less stuff outside the cell, water wants to move where there's stuff at, and there's more stuff inside the cell, right? Water will rush into the cell will basically burst and then lyse, right? And so it will become really, really large, okay? Remember the three types of endocytosis. Again, this is bringing something in the cell. So we have phagocytosis. Notice how the cell membrane, it kind of wraps around that material and it engulfs it. Again, we're using ATP. So phagocytosis is cell eating, right? So we're bringing in solid material. Penocytosis is where it's basically cell drinking. So we're bringing in liquid material. And then we have receptor-mediated endocytosis. And so remember that. This is where that ligand will bind to a receptor protein and it induces its endocytosis inside the cell in a vesicle. Okay? Also, remember osmosis. So whenever we're talking about osmosis, remember we're going to be talking about water, the movement of water. So let's just look at it first. Osmosis is the movement of water, or I should say the diffusion of water from an area of high water concentration to an area of low water concentration. But because we know that water wants to move where there's stuff at, we can also say osmosis is the diffusion of water from an area of low solute concentration to an area of high solute concentration. Okay, so if we look at this example right here, you want to be able to explain the direction of movement of various things, right? So water wants to move where there's stuff at. If you calculate like the amount of stuff, basically. So for example, inside the cell, this is like inside the cell, right? So we have, 9 plus 4 plus 10, that's like 23, percentage of stuff, basically. Outside the cell, we have 40 plus 10 plus 10, that's 60% of stuff, basically. So, right, water wants to move where there's stuff at, right? So water is going to move out of that sac into that extracellular space, basically. When we're talking about glucose, right, so we have 10% glucose outside, 9% glucose inside. Glucose wants to move down its concentration gradient, so glucose will want to move in, right? Water, we know, moves by osmosis, okay? And albumin, it actually like, so for example, what about albumin? Well, we know that the sac is permeable to all substances except albumin. So albumin will not move because it won't go in or out because that sac is not permeable to albumin, so it won't move, right? So remember these different properties, okay? Happy studies.